This protocol provides a framework for studying the incorporation of exogenous fatty acids from complex host sources into bacterial membranes, particularly Staphylococcus aureus. The protocol utilizes unbiased lipidomic analysis and lipoprotein particles isolated from chicken egg yolks as an economical source of complex fatty acids to quantify fatty acid incorporation into bacterial lipids.
The mass spectrometry techniques described here offer an unparalleled perspective of the fatty acid profile of Staphylococcus aureus lipids, but can be adapted to study other bacterial membranes.
When performing this protocol, care should be taken at each fractionation step to identify and retain the LDL-containing fraction. Removal of the ammonia sulfate from the plasma fraction is critical for downstream applications of the LDLs. Provide ample room in the tubing and change the water as needed to permit effective dialysis.
To begin, wash 2 egg yolks twice with 30 milliliters of sterile phosphate-buffered saline to remove residual albumin. Puncture the vitelline membrane with a sterile pipette tip and drain the contents of the membrane into a sterile 50 milliliter conical centrifuge tube. Add approximately 24 to 30 milliliters of 0.17 molar sodium chloride solution at pH 7 to the egg yolk and mix vigorously. Then place the tube on a rocking shaker to mix at 4 degrees Celsius for 60 minutes. Centrifuge the egg yolk dilution at 10 degrees Celsius at 10,000 times G for 45 minutes. Transfer the supernatant plasma fraction into a sterile 50 milliliter conical tube, leaving behind the pelleted granular fraction.